A very good morning, children, and welcome to Science Online class. So, from today, we are going to begin with a new lesson from Science 2, that is, lesson number 8, Useful and Harmful Microbes. Before starting with the lesson, let's answer some questions. What is meant by microbes? So, students, do you know what are microbes? It is a microorganism, especially a bacterium causing disease or fermentation. Some microbes are useful and some microbes are harmful. So, what is meant by microbes? A microorganism, especially a bacterium causing disease or fermentation. Next is, what are their characteristics? Now, what are the characteristics of microbes? How they are? Some microbes may be unicellular or multicellular. They may be of single cell or made up of many cells. So, they may be unicellular or multicellular. They can't be seen with naked eyes but are visible by microscope. They are found everywhere. They are useful at many sites and harmful too. So these were the some characteristics of microbes. Next question is, how did you observe microbes? So microbes can be observed under a microscope. Now let's see some of the useful microorganisms. The first one is lactobacilli. Here one activity is given. Let's see what's the activity. Smear a drop of fresh buttermilk on a glass slide. Stain it with methylene blue and put a cover slip over it. Observe the smear under the 10x objective of a compound microscope and then with the more powerful 60x objective. Did you notice the blue rod-shaped organisms moving around? They are lactobacilli, a kind of bacteria. So students, what is the activity? We have to take a drop of fresh buttermilk on a glass slide. Then after that, we have to stain that drop with methylene blue and put a cover slip over it. Afterwards, we have to observe it under the 10x objective of a compound microscope, and after observing under 10x, then we have to observe it under more powerful objective, that is 60x objective. You will see that the blue rod shaped organisms moving. So that blue rod shaped organisms are the lactobacilli bacteria. Lactobacilli is a bacteria. They are minute and rectangular in shape. What is minute? Minute means very small. So they are very small and they are rectangular in shape. Lactobacilli are anaerobic bacteria. And what is anaerobic bacteria? What is anaerobic? That is, they can produce energy without the use of oxygen. Organisms that can produce energy without the use of oxygen are said to be anaerobic. So, lactobacilli are anaerobic bacteria, that is, they can produce energy without use of oxygen. So, what we come to know, students, of lactobacteria? Lactobacilli are blue color rod shaped organisms. Lactobacilli is a kind of bacteria. They are minute and rectangular in shape. Minute means they are very small and they are rectangular in shape. Lactobacilli are anaerobic bacteria, that is, they can produce energy without the use of oxygen. There is one more question, and it is, how is yogurt made from milk? What exactly happens in this process? Have you ever think about it, students? How is yogurt prepared from milk? What exactly happens? So let's see. The lactobacilli convert lactose, that is the sugar in the milk, into lactic acid. This process is called fermentation. As a result, the pH of milk decreases, causing coagulation of milk proteins. Thus, milk proteins are separated from other constituents of milk. This is what happens when milk changes into yogurt. Yogurt has a specific sour taste due to lactic acid. The low pH destroys harmful microbes present in the milk. So, exactly what happens, students? To turn milk into yogurt, these bacteria, the lactobacilli bacteria, ferment the milk, turning the lactose sugars in the milk into lactic acid. So, the lactose sugars which is present in the milk, it is turned into lactic acid. The lactic acid is what causes the milk as it ferments to thicken and taste tart. Because the bacteria have partially broken down the milk already, it is tough to make yogurt easier for us to digest. So, what happens, students? Exactly what happens? Bacteria ferment the milk, that is turns the lactose sugar into lactic acid, and the lactic acid is what causes the milk as it ferments to thicken and taste tart. So, this process is called fermentation, and as a result the pH of milk decreases, causing coagulation of milk proteins. What is coagulation, children? Coagulation means to make milk thicker, to make the clots. That's why curd is more thicker than the milk. Then milk proteins are separated from other constituents of milk. This is what happens when milk changes into yogurt. Yogurt has a specific sour taste due to lactic acid. The low pH destroys harmful microbes present in the milk. This is how actually the process of converting milk to yogurt, and this is done by the bacteria named lactobacilli. So, lactobacilli is said to be a useful microbe because it helps to convert milk to yogurt. That's why it's a useful microorganism. Moving to the next microbe, it is Rhizobium, symbiotic bacteria. First of all, why Rhizobium is said to be symbiotic bacteria? Do you know the meaning of symbiosis? Symbiosis is a term describing any relationship or interaction between two dissimilar organisms. So, students, let's see how Rhizobium interact with other, hence it is said to be as symbiotic bacteria. Now, here's one activity given. What's the activity? Let's see. Take a plantlet of fenugreek, groundnut or any other bean and sterilized it with a 3 to 5% solution of hydrogen peroxide. Then afterwards, keep it in a 70% solution of ethyl alcohol for 4 to 5 minutes. Clean the roots with sterile water and take thin sections of root nodules. Select good section and place it in a solution of safranin for 2 to 3 minutes. Place the stained section on a glass slide. Cover it with a cover slip and observe it under the compound microscope. The pinkish rod-shaped organisms are Rhizobium bacteria. So students, what's the activity? We have to take a plantlet of fenugreek, groundnut or any other bean and sterilize it with a 3 to 5% solution of hydrogen peroxide. After sterilization, we have to keep it in a 70% solution of ethyl alcohol for 4 to 5 minutes. Then we have to clean the roots with sterile water and take thin sections of the root nodules. Do you know, children, what are root nodules? So, a swelling on a root of a leguminous plant containing nitrogen fixing bacteria are said to be root nodules. Then we have to take thin sections of the root nodule Then select good section and place it in a solution of safranin for 2 to 3 minutes. Afterwards, place the stain section on a glass slide. After staining the section of root nodule, we have to place it on a glass slide. Then cover it with a cover slip and observe it under the compound microscope. What we will observe, children? The pinkish rod-shaped organisms we will see. So what are those pinkish rod-shaped organisms? They are the rhizobium bacteria. So the pinkish rod-shaped organisms are the rod-shaped bacteria. So what we come to know about rhizobium symbiotic bacteria? That they are pinkish in color and they are rod-shaped. What is the role and importance of rhizobium? Rhizobia living in root nodules supply nitrates, nitrites and amino acids to that plant and in exchange get energy in the form of carbohydrates from it. Such a mutually beneficial relationship is called symbiosis. So students, this rhizobia bacteria which lives in root nodules, what it do? It supply nitrates, nitrites and amino acids to that plant and in exchange it get energy in the form of carbohydrates from the plant. So such a mutually beneficial relationship is called symbiosis. Do you know what is mutualism? Mutualism is a symbiotic relationship between two organisms where both the organisms are benefited. So is it correct, children? Here what does rhizobacteria do? It provides nitrates, nitrites and amino acids to the plant and in exchange it takes energy from the plants in the form of carbohydrates. Means both the organisms are benefited over here. Plant is also benefited and the bacteria is also benefited. So such a mutually beneficial relationship is called symbiosis. Rhizobia produce nitrogenous compounds from atmospheric nitrogen. However, for this process of nitrogen fixation, they need leguminous plants like beans, sweet pea, soya bean, etc. as host. Beans and pulses are rich in proteins due to the nitrogenous compounds made available by rhizobia. So rhizobia produce nitrogenous compounds from atmospheric nitrogen. From the atmospheric nitrogen, that bacteria takes the atmospheric nitrogen and it produces the nitrogenous compounds. So for this process of nitrogen fixation, it needs leguminous plants. What are leguminous plants, children? Leguminous is used to describe plants in the legume family, which includes the plants that produce some beans, peas and lentils. Crops which have nodules in their roots which contain nitrogen fixing bacteria are called leguminous crops. So here the process of nitrogen fixation is going on. So it requires only leguminous crops. So after that, beans and pulses are rich in proteins due to the nitrogenous compounds made available by rhizobia. Why beans and other pulses are rich in proteins? It's because only due to the nitrogenous compounds that are made available by the rhizobia bacteria. After harvesting a leguminous crop, the leftover roots and other plant parts are deliberately dumped in farm soil to maintain its bacterial population. The use of rhizobium has helped to reduce the use of chemical fertilizers and thereby their adverse effects. It has also helped to reduce expenses on fertilization and thus works to the benefit of farmers. So once the harvesting is done of leguminous crop, whatever are the leftover things like leftover roots and other plant parts, they are dumped in farm soil. And why they do so, students? Why the leftover roots and other plant parts are dumped in the farm soil? To maintain the bacterial population. So the use of rhizobium has helped to reduce the use of chemical fertilizers and thereby their adverse effect. Using rhizobium has reduced the use of chemical fertilizers. It has also helped to reduce expenses on fertilizers and thus works to the benefit of farmers. So this rhizobium bacteria has benefited the farmers by reducing the expenses of fertilizers. So yes students, is it a useful bacteria? Yes, rhizobium, that is the symbiotic bacteria, is a useful microbe. Now next microbe we are going to learn, that is yeast. So there's one more activity. So what's the activity? Let's see. Bring active dry yeast from the market. Mix a spoonful of yeast, 2 spoonfuls sugar with a sufficient quantity of lukewarm water in a bottle. Fix a colorless transparent balloon on the mouth of that bottle. What changes do you observe after 10 minutes? So students, what is the activity? First you have to bring the active dry yeast from the market. Then with a spoonful of yeast you have to mix 2 spoonfuls of sugar. In that you have to add lukewarm water in a bottle. Next, fix a colorless transparent balloon on the mouth of that bottle. Now what changes are we going to observe after 10 minutes? Mix lime water with the gas accumulated in the balloon. Whatever gas is collected in the balloon, in that we have to mix lime water. So collect that lime water in a beaker. Then afterwards we have to collect that lime water in a beaker and observe it. What do you notice? Take a drop of the solution from the bottle on a glass slide, put a cover slip over it and observe it under the compound microscope. Then the solutions which we have prepared of the dry yeast, 2 spoonfuls of sugar and lukewarm water, that solution, from that solution we have to take a drop on the slide. Then what we are going to observe? We will be seeing the colorless oval cells of yeast on the slide. Some of those cells may have small round bodies attached to them. These are new daughter cells of yeast in the process of formation. So students, what we are going to observe? We are going to observe the colorless oval cells of yeast. They are the cells of yeast. Some of those cells have small round bodies attached to them, and that small round bodies are the new daughter cells of yeast in the process of formation. So this method of asexual reproduction is called budding. So students, this is the method of asexual reproduction and this method is called as budding. Yeast is a heterotrophic fungal microbe that grows on carbon compounds. Yeast is a unicellular fungus with 1500 different species in existence. The yeast cell is a eukaryotic type of cell. Now students, yeast is a heterotrophic fungal microbe. What it means? It is said to be as heterotrophic fungal microbe because it derives energy from organic compounds. And what are organic compounds, children? Compounds that contain carbon-hydrogen bond are said to be organic compound. So here yeast is a heterotrophic fungal microbe that grows on carbon compounds. It is heterotrophic, that is going to derive energy from organic compound, and yes, organic compounds are the carbon compounds. Now yeast is a unicellular fungus. Unicellular means made up of single cell and 1500 different species are in existence. The yeast cell is a eukaryotic type of cell. That means it has a well-defined nucleus. What are eukaryotic cell? Eukaryotic organisms are the organisms which have a well-defined nucleus in them. So the yeast cell is a eukaryotic type of cell. Now in the above experiment, yeast grows and multiplies very quickly due to the carbon compounds in the sugar solution. In the process of obtaining nutrition, yeast cells convert the carbohydrates in that solution into alcohol and carbon dioxide. This process is called fermentation. Now what is fermentation? We have seen in the above experiment that yeast grows and multiplies very quickly due to the carbon compounds which are present in the sugar solution. And the process of obtaining nutrition, yeast cells convert the carbohydrates in that solution into alcohol and carbon dioxide. So what they do? There is the conversion of the carbohydrates into alcohol and carbon dioxide. So this process where there is the conversion done in the process of obtaining nutrition, so this process is called fermentation. So students, from all this what we come to know that yeast is also a useful microbe because it's helpful for fermentation. The bakery products are made by fermentation. So they use this bacteria for fermenting. So this is the useful microbe. So students, this was all about today's lecture. What we learned? First, we learned what are microbes, what are their characteristics and how to observe microbe. Then we started learning some useful microorganisms. In that, first we learned lactobacilli, second rhizobium, which is said as symbiotic bacteria, and third one yeast. In our next lecture, we will be learning about another concepts. So, students, watch the video attentively to get all your concepts clear. Thank you for your attention. Goodbye. Have a beautiful day.